Hi, A&Pers. This is Dr. Jones checking in. This is A&P II Lab 4, video 7, if I'm keeping track right. So we are here with the embryology model set, and we're going to be covering this last part, page 36, if you are playing along at home. And on this, see how we've got in the notes the Model 1 ovum and sperm penetration. And on here we have, this is Model 1. And then here it says structure number 1. Oh, here's a 1. This is, in fact, the corona radiata. And number 2 is the zona pellucida. And number 3 is the cytoplasm of the oocyte. Number 4 is the nucleus of the oocyte. And the number 5 on that dark blue there is, in fact, the sperm. So on these models, gasp, the numbers are in our book. Yay. Okay, so we are going to go through these together. So let's go through model number 1. And again, the corona radiata are the cells that surround the oocyte. 2 is the zona pellucida, which is a membrane that surrounds the oocyte or oocyte. The cytoplasm is number 3. The nucleus is number 4. It kind of looks like, you know how Easter eggs, when you like dye them and the dye goes into the egg a little bit? That one just looks like an Easter egg to me. And then, of course, the sperm, and this is the head of the sperm, and that's the string is the tail. And so it is entering the corona radiata. So this is what, as the sperm starts to penetrate the oocyte, the ovum. Okay, we're at model number 2 now. So this is fertilization. So the sperm has entered the oocyte, and that causes oocyte activation. So it is activated, and that changes the membrane. We'll talk about that in lecture. And that prevents polyspermy. It prevents another sperm from entering, and usually. So we still have the zona pellucidum, and then that, see that little white line there is the edge of the oocyte, so that'll be the fertilization membrane, and then of course the cytoplasm and the nucleus. Model 3. Model 3. This is pronucleus formation. So this cell now completes meiosis and becomes an ovum. So it's released as a secondary oocyte, and it does not complete meiosis to become an actual ovum, an egg, until if it is fertilized. So these are the chromosomes, these dots are the chromosomes of the oocyte. Number 2 here are the spindle fibers. We've got the male pronucleus, that's the one that looks like a Halloween skull, with red glowing eyes. And the polar body are the little marshmallows off to the side, and I'm talking about the little dried marshmallows that come in the hot chocolate. So those are your polar bodies. Model number 4, pronuclei migration. So the female pronucleus is blue, and the male pronucleus is pink. Now normally now our colors are switched, where we say boys are blue and girls are pink. I forget how long ago. The colors were actually the opposite, because pink was considered a masculine color, because pink is red, and red is a strong color. And then baby blue is a delicate color, and so that's clearly more feminine. And then a different clothing company had a better marketing campaign, and therefore our colors switched. So that's why pink is for girls and blue is for boys, because someone had a great marketing campaign a long time ago. So there's that. Excuse me. So this is model 4, and we have the 2 little marshmallows, which are the polar bodies. It says in the thing first and second polar body, but we don't care which is which. We just know that there are polar bodies. Model 5 is amphimixis. Well, this model is just prior to amphimixis. Amphimixis are when the two pronuclei fuse. So you do need to know that term. Amphimixis is the fusion of the two pronuclei. So the female pronucleus is still blue, the male pronucleus is still pink, and now we have the centrosomes with centromeres in them, and marshmallows are polar bodies. Model number 6, and this is a zygote. So the male and female pronuclei have fused. This is a zygote. So if it says name these, name of this phase of my phase, this is a zygote. It is diploid. It has a full number of chromosomes. It has 23 pairs of chromosomes, so 46 total. And of course, the marshmallows are polar bodies. Let's see, model 7. First cleavage, and you need to know that term, when the zygote splits in half, and when this divides, this is no longer meiosis. Meiosis is when we made the egg and when we made the sperm. And this cell division does not make an egg, it does not make a sperm, so this cannot be meiosis. We are back to mitosis, and we won't get to meiosis again until we make eggs and sperm, which in the case of people with ovaries, you make the eggs before you are born, and people with testes don't make sperm until after puberty. So when this zygote divides in two, that is mitosis. And like when you step on your sister's toe, maybe she steps on yours. Mitosis. I know, it's terrible. You're welcome. So this is the cleavage, is when the mitosis splits the zygote into two cells. So each of these cells is a blastomere, and you need to know that term. You need first cleavage, and you need blastomere. So the cell's a blastomere, and that cell's a blastomere. And of course, you've got polar bodies. Okay. 8. Move these forward. These are next. Okay. That one's acting weird, but we're good with that. So this is number 8, and we've got our second cleavage. So each cell is still a blastomere, and we still have polar bodies. Number 9. This stage is called the morula stage, and that means raspberry. So it's just a clump of cells, a ball of cells. Each cell is still a blastomere. We have the nucleus, the cytoplasm, polar bodies, morula. Okay, so 10 is where things get interesting. This is model number 10. So now we have implantation. So this is our fertilized oocyte. This is the morula. Actually, at this point, it's a blastocyst. And then this is the wall of the uterus, the endometrium. So this would be the endometrial epithelium. And this is the endometrium. So 7 at the bottom of our list here is the endometrium of the uterus. 8 is the uterine epithelium. Okay. So this hollow ball of cells is a blastocyst, and we need that term. Blastocyst forms day 7 or 8, hollow ball of cells. The trophoblast is the outer layer, need that. The inner cell mass becomes the blastodisc, and that's what becomes the embryo. So there are 2 parts of the trophoblast, the part that maintains its cell membranes and nuclei. This is the cellular trophoblast. And then this part is the syncytial trophoblast, and that's number 6. I'm not going in order anymore, am I? Okay. So number 1 is the cellular trophoblast. Number 2 is this space, and it's a fluid-filled space. There's not air here. This is the blastocele, C-O-E-L-E, is often used for a cavity. The hypoblast is number 3, hypoblast, and 4 is the epiblast. And these are the 2 layers that are the blastodisc, which turn into the embryo. And then we've got the amniotic cavity, is that tiny little space right there, just big enough for my fingernail. Amniotic cavity, it gets bigger and surrounds the embryo. And then, as we said, syncytial trophoblast, endometrium, and the uterine epithelium. And that was 10. Now let's talk about 11. This is where things get interesting. So let's talk about the embryonic membranes. First, that epiblast has turned into ectoderm. So this is this flat layer of pink cells with light blue nuclei. So this is the ectoderm. And above it, this space is the amniotic cavity. As I said, it's getting bigger. 3 is the endoderm, and that's from what used to be the hypoblast. And 4, this space below it, is the yolk sac. Because you had a yolk sac, just like a chicken, and you used the nutrients from the yolk sac to grow, just like a chicken, because chickens don't grow in the yolk, they grow in the white. They eat the yolk, just like you ate yours. And so this next space, number 5, is the blastocele. 6 is the mesoderm, although the mesoderm is formed right here between the ectoderm and the endoderm, so it's weird that it's not shown there, but okay. So the sage color stuff is all mesoderm, although it should be there also. Skipping 7, 8 is the extra embryonic coelom. 9 are the chorion, and you see the little 9 right there. Okay, and we're forming little villi. And then 10 is the maternal blood sinus. So you've got 12 uterine arteries bringing in blood that's pooling in the maternal blood sinus. And then the, I think we're still at pre-embryo stage here. Well, maybe embryo. And so this is getting nutrients from the maternal blood sinus and putting wastes into it, and the wastes are carried out the uterine veins. And then these chorionic villi, in the area with the most nutrition, so where the best blood supply is, will develop into, eventually, the placenta. Model number 12, let me turn the page. Okay, so here we have an embryo at 3 weeks. So this double layer right here now has 3 layers, and then it folds. So this part is the head fold right here. So that's what is going, it's turning into the head slowly. And this green stripe right here is the notochord. And the notochord becomes your spine and all of that. In fact, there are remnants of notochord in your intervertebral discs as the nucleus pulposus. Number 4 is the chorion. 5 is the amniotic cavity. Again, it's getting bigger, going to surround the embryo. The yolk sac, still yellow, and that is the tiny little heart. And technically they're like heart type. It's not a heart like with 4 chambers like we learn in cardiovascular, but it is sparking and moving blood. So it's not quite a heart, but it's getting there. And the body stalk is, again, not an umbilical cord yet, but moving that direction. So this is the body stalk. And then the chorionic villi are not a placenta yet, but moving that direction. Okay, next model is model 13. And this is the embryo at 4 weeks. We've got a yolk sac. And 2 here is the body stalk. Again, not quite an umbilical cord yet, but working on it. 3 is the amniotic cavity. The amniotic cavity is the space. The amnion, number 4, is this white membrane. So the cavity has the amniotic fluid in it. The amnion is the membrane that surrounds and contains that fluid. Number 5 is the chorionic cavity, which is this space here. Later, the chorionic membrane will meet the amnion and they'll become the amniochorionic membrane. So if you've ever had your membrane stripped, it was the amniochorionic membranes that they were talking about. The decidua capsularis is the shiny part, and that's the part that sticks out into the uterine cavity. And the decidua bassalis is the rough part. That's the part that is in the endometrial layer. So as you can see, the chorionic villi are bigger in the decidua bassalis because that's where the blood supply is. In the decidua capsularis, there's less blood supply. The chorionic villi are kind of degenerating, and the placenta will develop where the blood supply is. We've got a maternal artery, maternal vein, and then here we have embryonic blood vessels. Okay, so those were models 1 through 13, the embryo up through 4 weeks. This is A&P II Lab 4, video 7 maybe. The education must go on. Dr. J out.